we're going to be talking about digestion, absorption of food, mostly macronutrients. You know what they are: carbohydrate, protein, fat, macronutrients. The references in Guyton are here. That's your textbook. But I realized that you also have Lippincott, biochem. Well, that material is covered very nicely in Lippincott. Right? It's only a few pages, and it's up to date. Okay, it'll give you up-to-date information on the topic. Your choice, though. Again, if you understand the PowerPoint, fine. If you don't understand the slide, or it's difficult to understand what's going on in the slide, go to either Guyton or Lippincott, and it'll make it clear for you. I hope. Okay. You had that biochem textbook, right?
Yeah.
Yeah. You know, there's not much storytelling there. It's just you facts of what's going on.
Which part? Micronutrients?
Pardon me?
Which part in the book?
Which
chapter?
book?
Which chapter in the book? Like micronutrients? I'll come right there.
Patch is 56. This, I gave you the page number. Carbohydrates, proteins, lipids, it's spread out. Same thing in Guyton. He does. I think in the middle here, he's covering electrolytes or something. We'll cover that next week. You can read it if you want, but it won't help you for these slides. Okay? But now you have two references you can go to. Or any other, doesn't matter to me, I told you before, any other technique. All right, a little review and introduce you to our concept, and hopefully you get a big picture of what's going on here. Is we already know about this a little bit, right? Stem cells, they're the ones that go through mitosis, they're usually located in the crypt. And then the cells we're going to be interested, the ones that are digestion, absorption, enterocytes. They're up here in the villi, in the intestine, right? They migrate up from the stem cells, differentiate, and they mature. So you can see it takes a while to become fully mature. It's just like you growing up or something. They are not fully operational until they're in the upper third of the villi. Okay. Now the upper third of the villi also is the one exposed to, I hope I wrote the word harsh environment. That's in the lumen. Remember in the stomach, those surface epithelial cells are exposed to acid and dust. That's what I call aggressive acid. Okay, it's going to hurt you and hurt these cells. And that's one of the main reasons these stem cells are so important. You lose those cells, and even though the stomach, for example, has mucus and secretes bicarb, nothing's perfect, right? Eventually, and these cells last 3, 4, 5 days before they finally go hepatic. And then apoptosis, and then we get replaced. It's the same thing in the intestine. What's in the intestine? The pancreas drops all these enzymes in, right? You have pancreas, right? Enzymes? They all go in there. You think those peptides, or those peptidases, or lipases, or any of those things, they care what they're hydrolyzing? They're not thinking about it. Fortunately, in other words, you're creating a whole soup of aggressive material, all right? A free-for-all, if you will, for the enzyme. And the one thing that's in your favor is that you offer them competition. Instead of eating you give them food. So when food's there, less chances of them interacting with your epithelium, they're going to interact with the food constituents, right? It's a free-for-all. And you'll notice if you don't have food, what about the sphincter of Oddi? Is it open or closed? Closed. You don't want to, why put pancreatic enzymes in there when, what do they have to do? The only thing they can do is eat you, right? I think you're getting a general idea, right? It's a harsh environment. Bile salts are also toxic from the liver, but that sphincter of Oddi is closed off. And the only time you get contraction of gallbladder, pancreatic enzyme secretion, opening the sphincter of Oddi is when the food's there, right? So, all right, that's the food. Is that okay?
Yes.
Just give you a general idea. Now, digestion, you know it's all hydrolysis. It's all hydrolysis, whether it's breaking a peptide bond, hydrolytic cleavage, whether it's breaking an ester bond on triacylglycerols, it's going to be hydrolysis. Okay, same thing with carbohydrates, hydrolysis. It can happen in the lumen, microvilli that are brush border. You're going to have pancreatic enzymes in the lumen, and you're going to have enzymes in the brush border. And in one case, only for protein, you're also going to have peptidases in cytoplasm of the enterocyte, okay? So that's why I have this. For lipid, the only thing that's going to be involved here are the pancreatic enzymes in the lumen digestion. Carbohydrates and proteins are going to involve brush border enzymes as well, okay? Now what I have here is, if you're in the brush border, this kind of makes sense, the catalytic site of these enzymes, or the active site, or whatever you want to call it, is it going to be located internally or externally? You have no interest in eating your own material, okay? So that's good. The other thing is, I mentioned the harsh environment, and even these enzymes will be attacked. These enzymes will attack each other. When you dump all your pancreatic enzymes, well, one, activation of the other enzymes requires trypsin, right? It's attacking other trypsinogens, right? So they're going to attack peptide bonds. And so what happens is that eventually you can, during the process of a meal, you'll be destroying some of these. It's not a problem. You resynthesize them. Resynthesis, remember DNA and protein synthesis and all that? That takes about, what, 2-3 hours? That's good, because that's how much time you have between meals. So whatever you damage on the membrane is resynthesized and put back up before the next meal usually. Does that make sense? This is different from reepithelialization or repair. That involves 3 or 4 days, and that's the entire cell moving up, right? This is a cell that loses some protein from the microvilli. It can upregulate. And this applies for transporters too. Transporters too can get damaged. They're proteins, right? They can get damaged. The same thing. We can upregulate these transporters between meals. Okay, let's take a look at carbohydrates first. They're the easiest ones to understand. They're going to involve both the lumen and brush border. Here's a key point, monosaccharides. That's the only way we can transport them, absorb them, as monosaccharides. Yet we don't ingest them that way, right? Starch, it's a polysaccharide, many. Sure. It's all glucose, by the way. And they have 2 types of bonds. You might well know them, I guess. Well, starch is a plant storage, and humans will store glucose on glycogen. It's the same kind of concept. And starches in potatoes, rice, things like that. Starch is composed of amylose and amylopectin. Most of it's amylopectin. And the reason we bring that up, because most of it is that you're going to be dealing with alpha-1,6 linkages. Glucoses, monosaccharides, these units are arranged, if they're linear, with alpha-1,4 linkage. So if they're unbranched and it's a straight chain, glucose is all going to be alpha-1,4. We have our pancreatic enzymes have no problem with that. Now, at the branch points, it's no longer an alpha-1,4 linkage, it's an alpha-1,6 linkage. You can't, well, blow it up. It's all PowerPoint. It's alpha-1,6, that's what that is at the branch point, okay? So it's a complex starch. The other ones are disaccharides. That means 2, okay? So we only have to cut them in half, so to speak, right? So this, you don't need the pancreatic enzyme for sucrose, which is sugar, or cane sugar. You put that in your tea, or comes in fruit, right? There's glucose and fructose, if you ever heard of that. Lactose, it's in milk and milk products, and it's also a disaccharide, okay? And you don't need the pancreatic enzymes for that. Brush border enzymes are pretty important. Ones in them, like real, because you can't absorb the disaccharide. You need a monosaccharide to be able to absorb it. All right. Cellulose is also like starch. It's all glucose. It's also called fiber. I don't know if you've heard of that, but cellulose is in plant wall. I always think of celery when I think of cellulose. So like a lot of fiber. And our pancreatic enzymes and brush border enzymes can't really deal with it very well because of the linkage. It's beta-1,4. So we can't Our pancreatic enzymes and brush border enzymes, or humans, we can't process cellulose very well because of its structure. We'll get to that in a minute. Okay. All glucose subunits, this is amylose, alpha-1 4 linkages, and here at the branch point, alpha-1 6 for amylopectin. The only difference is one's branch, one's not branch. And here's a convenient thing taken from somebody else's textbook, but alpha-1 4 linkages will be cut by pancreatic amylase. Now, amylase is a, where is that? Endoglucosidase. What is that? Endo. Remember endopeptidase is exo? It can only cut, it cannot cut from the terminal end of either one. So the most you're going to get out of this, if the pancreatic enzyme amylose is very effective, is two glucose units or 3 glucose units. The 2 glucose units together are called maltose, something you have to memorize, I guess. But once you have maltose, then the 3 become maltotrials, tri meaning 3, right? So that's not too hard, is it? No. Because we can't get in, pancreatic amylase can't get injured, so then it becomes an exo, right? Rather than an endo. So glucose is not a product or a monosaccharide. You're not going to get a monosaccharide just using pancreatic enzymes, okay? So you know something more has to come, right? Now with alpha amylase working on amylopectin, it's no problem. You're going to get the same thing, maltose trials and maltose, no problem. But you can't cut this alpha-1 6. These things are called dextrins. Depending on the textbook you read again, alpha limit dextrin, starch dextrin. But once you see the word dextrins in there, you know what it is. It's going to have a branch, alpha-1 6. Okay. So now we're stuck with these. We need to break them down to monosaccharide. And that's the job of the brush border. So let's take all this down to the brush border. This is the same slide from before, right? And there's your maltotrials, alpha-limit dextrin, maltose, and I got this thing in here. If you cut that alpha-1 6, what do you get? Maltose and maltotrials. Perfect. Let's take a look at it. Here's alpha-limit dextrin. And again, name changes. It used to be called dextrinase, good name for it, but most textbooks now will refer to it as isomaltase. And you'll see why in a minute, I hope. But that's what it's called now, isomaltase. But again, if this shows up on some kind of an exam or something, we're not going to leave it alone. I think Guyton refers to it as dextrinase, so if you're reading that textbook, you'll see isomaltase in parentheses. We're not out to fool anybody, okay? So, the product's going to be maltose and maltotrials, right? Perfect. What handles maltose and maltotrials from here? Wow, maltase, right? That's hard to remember. So in essence what you got an enzyme called maltase. It'll take all the maltose, maltotrials down to glucose, right? So it's all glucose units. Over here with the alpha-limit dextrin, isomaltase works first, then back up here. Okay? Straightforward. Now, sucrose is handled by a disaccharidase in the brush border. Guess what the name of that enzyme is? Sucrase. So that's not too hard to remember. Sucrase will attack sucrose. With lactose? Lactase. Okay, we're on it. This is easy, right? A few things to memorize, but pretty much easy. And you'll notice when we talk about amylopectin, that's all glucose. And amylose is all going to be glucose. Sucrose is a combination of glucose and fructose. Lactase is glucose and galactose. End product, primarily glucose. 80% of what's going to happen when you eat is going to be glucose. And guess what? You spend the most time on and metabolizing in biochemistry. Glycolysis, remember that? Probably don't want to remember it, but that's why you don't spend a lot of time with the proteins and the glucose. You don't spend much time with galactose and fructose. Okay, glucose is the predominant one. All right. So now we've got glucose. That's monosaccharide. We can absorb that, and the way we absorb that is using transporters. Because these are water soluble, you have to get them across the plasma membrane, right? SGLT1. Guyton doesn't bring it up in the GI section, but they have SGL, he does in the kidney. They have SGLT1 in the kidney, SGLT2, okay? And you'll get, all these transfers we're going to be talking about for the next couple of weeks, you're going to get all the same ones in the kidney, okay? So learning them is not an exercise in futility. It's going to help you later. It'll come back to you later, help you in the renal section. All right, let's take a look at this. We know sodium ATPase, right? Sodium goes out. This is a primary active transport, right? That's where you learned it? Yes. And because sodium is pumped out, or transported out actively, you have, this becomes negative inside, right? And less sodium. So the gradient, it's called electrochemical gradient for sodium, is strong to pull it in, right? And so this gradient created by this creates a secondary active transport, and that's sodium coupled to glucose or galactose. SGLT1, easy. Sodium, glucose, transport. Okay? Sodium, glucose, transport. SGLT1. And it'll transport either glucose or galactose, because if you put them in together, they compete for that transport. All right, so you can move glucose and galactose in here. That takes care of that. What about fructose? Well, I think you've heard of glutes. Those are glucose transporters. Now, there's a whole, I don't know, there's about 6, 7 glutes. You've had GLUT4, that's one that's insulin dependent, right?
Yeah.
You've had all these glutes before, right?
Yeah.
Yes. Right. Probably in biochem, huh? Yeah. Anyway, GLUT5 transports fructose. This is not sodium dependent. Okay, it's called facilitated transport, if you've heard that term before.
Yes.
As long as there's a higher concentration of fructose on this side, this transport will take it across. Here, this transporter needs both sodium and one of these monosaccharides attached to it, or it doesn't move, right? You know that one, right? No?
It does not require energy, does it?
Now you've got glucose, galactose, and fructose inside the cell. You use GLUT2 to move it out.
Does it require energy, the SGLT?
The only one that requires energy is this one. This one requires energy, but only because this one's creating an electrochemical gradient.
Oh.
Okay? And it's a concentration difference. You got more fructose on this side, it's going to move in. All right. And I have some notes, and this is kind of leads you through it, I guess. All right, okay. I said at the bottom here, colon cannot absorb monosaccharides. It also has, but it has some help. We have some help here. You notice the word colonic bacteria. You ever hear the word microbiota? It's a big area now. As a matter of fact, you can buy things called probiotic things to help your microbiota. People are looking, they even have a microbiome now, which is a genetic analysis of all that. And with the thousands of species of microbes that we have harboring in the GI tract, it's a difficult area, but they're linking into various pathologies, okay, things that go wrong. They call it dysbiosis. But anyway, don't worry about that, but sometimes you'll see the word microbiota, they're referring, they're talking about the GI tract. A cute thing I ran across that may or may not be true, and I don't know who counted all this, but they say that all the bacteria you harbor, that is in your eyes, in your nose, mouth, lungs that you breathe in, skin, and the GI tract, you have 10 times more bacteria on you than you have your own cells.
Yeah, that's too much.
You go, okay, even if they're wrong about 10 times, even if it's the same, think about it, that's a lot of bacteria. And you live with it all every day. It's fine, right? And actually, the colonic bacteria here help us out. Now, this is a profile. This is a log concentration. And all that means is that one is 100, 2 is, well, 1 is 10, 2, 100, 1,000, up, factor of 10 all the time. So there's very little in the stomach, upper small intestine, okay? And then when you get to the level of the colon, it shoots way up. Okay, so most of our bacteria in the GI tract are in the colon. We know we have some in the stomach. Some of us have H. pylori, and there's other species. There's a multitude of species. And I'm just giving here some of the You don't have to memorize these. It's just an idea of why. Remember in the stomach, pH 2. Not very many bacteria are going to survive along with the pepsin, right? But here, when you get down to the level of the ileum colon, you're around pH 7. They like that, right? Bile salts are toxic. I don't know if Mohammed brought that up, if you have a lot of bile salts, and if they're not in my cells, they're detergents. They will interact with your membrane just like they'll interact with lipids, right? They don't care. They're empathetic. We'll get to that. Well, anyway, and there's very little bile salts here. If they do end up in here in the colon, then you usually get a little bit of diarrhea because they're injuring your epithelial bile salts, okay? Faculty, I have aerobes here, anaerobes here. And the reason for that is to attract attention that they don't have actual measurements for it, but the PO2 means partial pressure of oxygen. It's higher here than it is here. Here there's almost no oxygen, it's almost anoxic. And so you'll see that the metabolism switches. Most of these bacteria in the colon are fermenters. They ferment rather than using glycolysis, okay? That's on oxygen. They don't have oxygen. So they're fermenters or anaerobes. They function in a low oxygen environment. Propulsive motor activity, remember gastric emptying. And then you have motor activity, we haven't covered that yet, we have motor activity, always moving things down. And then you have the MMC, the migrating motility constant. One of the strongest contractions, takes everything here and pushes it down to the terminal ileum into the colon. So everything's being moved to the colon. And here the motility is designed to just slowly take up all the water that's left. You want to save all that water, okay? Anyway, colonic bacteria. They salvage non-absorbed nutrients, okay? See that? 30 to 40%. I like that. Dry weight, okay? Some textbook routes say 30 to 40% of your stool is bacteria. Think about that next time you go to the bathroom. 30% of that, that's a lot of bacteria. Most of it's dead, you know. And it gets wiped out with the mucus and with the fecal matter. It sounds interesting, or everybody smiles, but that's the fact, okay? Most of it's going to be dead bacteria. And in essence, one of the major ways of perpetuating an infection is anal to oral. You haven't had microbiology yet, the bug stuff. That's the way disease from diseases are spread. That's why they want you washing your hands in the restroom after you're done. Okay? All right. They salvage nutrients for us. How do they do it? Well, for example, cellulose. We can't absorb it. We don't have enzymatic machinery to take it down to monosaccharides. Bacteria ferment. This is not hydrolysis, cutting glycosidic bonds. This is fermentation. And they'll generate small, acetic is 2 carbons. So they're taking, well, cellulose is long, but let's say you just had glucose in there. That's 6 carbon. They're going to be converting it to 2 carbon entities, 3 carbon entities. It's fermentation. And these are, acetic will be 2 carbon, propionic 3, butyric probably 4. And they all can be absorbed by the colon using a sodium covalent. They don't have names for this yet, so we don't give it. But they're absorbed into the sodium. Why? They're water soluble. I know they're called fatty acids, but they're really short chain. Okay? We'll come back to that later. And butyrate, especially, and all the other ones, they're even used by the cells in the colon as an energy source. And we can use them as energy sources, recycling. So we salvage what we can't, the bacteria take care of us in a way. We salvage some of the nutrients, yeah?
The sodium co-transporter in the colon, is it just for short-chain fatty acids?
Short-chain fatty
Nothing
acids.
else.
You don't have transporters for amino acids, you don't have transporters for glucose, you don't have, yeah. Anyone mentioned to you not, but most of your meal is digested halfway down your jejunum, you know, most of the nutrients. Bile salts, certain other things are saved or salvaged in the terminal ileum. You got that transport mechanism. But very little nutrients show up in the colon. And what nutrients do show up, bacteria take care of it. They ferment it. And one product is gas. Everyone likes hearing about this. Google it if you want. This slide, actually, I have to cut this off. Because it said the title don't take offense, okay, I'm just making a joke. The slide said, gas composition of farts, okay? Did you ever hear that? It's colloquial, what do you call it, street talk, right? I don't know if you've ever heard it, but it's actually the composition of flavors. That's the terminology you have to use, flavors. But anyway, gases are produced because of that. Why am I bringing this up? Because we're going to take advantage of this. We're going to be clinicians, right? No, hey, you take urine samples, fecal samples, right? You use a stethoscope, which is simply an amplifying machine to listen to things. You're going to take advantage of these gases. Here I just say fibers because that's another slide, but fiber is another word for cellulose kind of thing. We'll get to that. The major ones form are carbon dioxide, methane, hydrogen. And you have a choice. You can either it gets into your blood and comes out of your lungs like carbon dioxide or hydrogen or something like that, or you pass it as a gas, okay? And this may sound funny, but most normal people will pass about 1 liter of gas a day. It's not more. Whether you know it or not, you'll be passing that gas as flavor. So the next time you're in an elevator, and especially if it contains some trace odorous gases, like hey, it's not me, it's my happy microbiota. They're happy and they're eating, right? Which is true. You had nothing to do with it. It's the bacteria. All right. Why do we care about this? You'll notice that most of it is
Nitrogen.
No, nitrogen. And then you have hydrogen. The next one is carbon dioxide. We can't use that clinically. And the next one is methane. All right, let's look at the deficiency, and I'll come back to this gas. Yeah. The lactase, that means you don't have that enzyme, or that enzyme is ineffective. Oh, go ahead. I didn't mean to interrupt. I'm tired to do this. Wasting my time. You waste your own time. Okay, anyway, you don't have lactase, or it's not very well, not very effective. There's different stages of intolerance. Okay, I'll get to that in a minute. Where you have lactose coming into the small intestine, you can't hydrolyze it. Okay, which means you don't form glucose and galactose, which is absorbed almost immediately by these transporters. Now you know osmosis, right?
Yes.
So basically, this is a little difficult to understand. Usually, wherever the more solutes are, that's where the water will stay, okay? In this case, with lactose, you're not allowing it to be absorbed, so it's retaining water, okay? So that's a hard thing to understand. It's not like you created more solutes. It's retaining the water, because if you would have broken it, solutes would have went across and water would have went with it. And this is important, you don't know that yet, but we'll get to that next week, is you have a lot of fluids. You have gastric secretion, the pancreas secreted, the liver secreted, salivary gland secreted. It's all in here, right? And you drink water, some fluid. So there's a lot of fluid coming into your intestine. So as long as you can move that fluid out, usually it follows solutes. All right, so now you didn't take care of lactose. You're holding on to more water than you would have ordinarily. You get down here, your friendly bacteria. They don't care if it's cellulose, lactose, sugar. They're fermenters. They're going to ferment that sugar. As a matter of fact, that's how they use fermentation to make alcoholic beverages. Sugar. You knew that. Did everybody hear that before? No? Yeast. They're good fermenters. Anyway, you're going to generate, as we know, small carbon units. Here they're showing you lactic acid, which is fine. And we have acetic acid, you have propionic acid, butyric acid. What they're really attracting your attention to is the gases, carbon dioxide and hydrogen. Nice, huh? Remember urease? Test for urease? You can take advantage of these gases formed by the bacteria, okay? We'll take a look at that. How do you do something like that? Well, guess what? You don't have to label the carbon like with urea. We don't make hydrogen. Just measure the hydrogen the person's breath, right? I'll show you that in a minute. Now here, what you end up with is a lot of water coming in, right? Bloating, I'll buy that. Diarrhea, maybe. Dehydration, hard to like. That's pretty severe, right? Now it could be, if you have a severe lactose intolerance, you could take any milk product and you're in basket, and you're feeling very uncomfortable. You get diarrhea, right? But you can have mild lactose intolerance. And you can take some milk, some whatever, without having to undergo that. Maybe you go, it'll go away, okay? It's all different levels. And the reason I bring that up is because we take advantage of it too clinically. And you've heard of dietary fiber product? In the West they have something called Metamucil. You buy it in a store. I don't know where you can buy it here, probably a grocery store, but it's fiber. Why do people use it? They use it for this specific reason. It's going to hold water, okay? We can't digest it very well. And primarily, very few normal adults have to use it, but when you get older, not like me, but older people, when they get older, your motility, your motor functions aren't as great in the intestine, all right? They have a tendency to develop constipation. So what do? Add fiber to the diet, holds the water in, stool passes salt, okay? Or a child or someone else, it's pretty safe to do, but it's taking advantage of this, but in a mild way, okay? It's just give it a little fiber, it'll hold water because it's not metabolized by you, and the stool passes nice and soft, okay? Because I don't know if you've had constipation before, but passing a hard stool is no fun, okay? So we use it, it's commercially available, they're called bulking agents. Let's go to the second hydrogen breath test. There you go, right in your office. What do you do with this patient? Make sure he has no food or anything. And then you give it, these 4 patients, you give them lactose. Guess what? If he or she is lactase deficient, they're not going to metabolize it, right, themselves. So it's going to eventually, there goes hydrogen. One of the products, okay? Now, if you notice it takes a lag time, it doesn't show up right away, and why is that? It takes time to get there, right? It's not going to show up right away. It's going to take time to get to the colon. Now, you can just Google hydrogen breath test. They'll have different pictures for you. Some cartoons will show it going up in an hour, hour and a half. Well, it depends on the transit time. Everyone has an individual transit time, but in your office you don't know that. Just have them breathe in there, and if it goes 2 or 3 hours or longer without any hydrogen, I think you're okay. The other one they're using, I'll show you in a minute. The other one they're using, I think, where's methane? This one, 7%. So now they're either hydrogen or methane. Hydrogen seems to be the cheapest one and most sensitive right now. But they're developing the test for methane as well. You don't have to label it either. We don't make methane. Yeah.
Do they have to be breathing in for like 2 hours straight with the breathalyzer?
You don't go.
For 2 hours though?
Huh?
For 2 hours?
Sample. Yeah, that's what you have to do. You have to wait a while. Okay, if you go, okay, I'll take a breath sample here at 30 minutes and send the patient home. What have you accomplished? Nothing. But, like I said, if 3 hours later they have it, then you go, at least we're not dealing with that. And this is a fairly common problem. I think, you know, you have lactose-free food or whatever it's called. You can buy this stuff. Or just stay away from milk products. There's another one, this is congenital, usually shows up in the pediatric population. And it's sucrose, what they call it, sucrase isomaltase deficiency. But it's really sucrose, that's the problem. Why is that? I didn't give it to you before, but now most textbooks will have it. Clinically, you run across it. This is actually a combination of sucrase and isomaltase, okay? That is because these two things are complex together in the breath border. Actually, they're one gene product. Yep, I got it here. They're one gene product, but they're hydrolyzed by our friend trypsin, okay? And they remain complex. This one gene product has 2 active sites. One's for sucrose, and the other one's for maltose, maltose triose, things like that. And that's shown here. You see the this is called alpha-limidextrin, if you will. And it'll chop it down to maltose, triose, and maltose. And up here you've got glucose and fructose. Which one is it? Green is fructose and sucrose. So we're familiar with how these work, but I presented them individually before. They're really located the same. So that's why it's called sucrase isomaltase deficiency. Okay, and this starts in babies when they come off mother's milk, and you have to substitute something, right? Usually it's fruit juices of some kind. You may not be familiar with this, but that's usually what they give them, some fruit juice. And that's when it shows up. Why? Baby starts crying, uncomfortable, a little diarrhea. Well, okay, what do you do to test whether this is a problem? Give them some lactose, right? Yeah, give them some sucrose. It's pretty straightforward, right? You give them lactose, you may not get anything. That's the problem. Give them sucrose. If hydrogen shows up, that's the problem. And all you have to do is stay away from some fruits or minimize the ingestion of fruits. This is a summary slide, real quick. We know all this already. Physiological symptom. All right. Malady. Okay. You got an osmotic load. You didn't absorb the lactose, cellulose, whatever. It's going to hold. Lactose is a good one. It's going to hold water. It's called an osmotic load. Here it gets to the colon. You're going to have fermentation, more osmotic load, right? You're adding to this, okay? Fermentation is also going to cause, that's a byproduct of gas production, okay? Now, with an osmotic load coming either way, you're going to increase the amount of fluid in the lumen. The gases and the fluid in the lumen cause distension. Okay? And distension, depending on the degree, it's going to cause pain. You may feel bloating. Distension is a good stimulus for peristalsis. And of course, you're going to make gas. Yeah.
So can we go back to the previous one?
Previous.
So we have a deficiency in saccharides, right?
There's a problem with this complex, but the one that's affected the most is sucrose.
So that's why.
But they're hooked together, so they call it a sucrase isomaltase deficient. There's something wrong with this complex. But the major problem is with the sucrose. It's not, you can give them starch, stuff like that, they're going to be fine with the maltose and maltose triose. But they're not going to be fine with sucrose. Okay? But they could call it, and probably did call it a sucrase deficiency a long time ago until they realized it was one gene problem. Okay? So that's the deficiencies in that gene. But it's, take my word for it. It exists as a complex, and that's why it's called proteins. All right, dietary sources, we know that, meat, eggs. And you have essential amino acids, non-essential amino acids. Some amino acids we can make, some you have to get from the outside, essential amino acids. But 50% of the protein material in your GI tract is surprisingly from you. It's endogenous. That's what the word means, it's from you. It's the cells that have sloughed off, dead bacteria, mucus secretion, mucoproteins, right? Goes back to proteination material, everything. Okay, so a lot of, and your own enzymes, if you think about it, what are they? The proteins. Okay. And as I said before, they're very non-discriminatory. They'll attack each other. It's not a problem. It's a very nasty environment. Okay. All right, protein digestion, we know some of this. In the stomach, remember, pH optimum for pepsin, 1 to 3. And you got the hydrogen there, so it's good for that. You're going to generate primarily, pepsin, remember, is endopeptidase. You're going to generate primarily oligopeptides. Maybe some proteins will get through. Now in the intestinal lumen, we have B. You cover this in the pancreas, right? Yep. All right. And you have bicarb. Now, it just so happens that the pH optimum, this is for trypsin, but all of them have a pH optimum around 7. That's just representative. So that's a good thing, right? And they're activated by something called, I got it in red again, another reminder, they're activated by enteropeptidase. Now, some textbooks still use the word enterokinase. I think probably Mohammed Ghazali brought this up. But that's been changed. I'll tell you what. You had Bio-Chem, right? What's a kinase? What does it do? It's phosphorylate something, right? What does this got to do with phosphorylation? You're hydrolyzing things. You're cutting a peptide bond. So they changed it from an enterokinase, which is which cuts a peptide. That's how you activate these things. You don't activate them by phosphorylating. Okay? They realized that whoever named it, they don't know why, but they're going, let's fix it. It's really an enteropeptide. But again, if it shows up on the exam, I know that some textbooks are still using
Enterokinase.
enterokinase, we'll have it in print. That's what we're trying to do that. But be aware of that, because in 2 or 3 years, by the time you're taking these, if you're taking any of these major international national exams, you probably won't see the word enterokinase anymore. All right, we have this as a summary. Pepsin, endopeptidase, no amino acids. Trypsin, chymotrypsin, endopeptidase, no amino acids, right? If you're an endopeptidase, and that's right here, little cartoon for you, the most you're going to do is create, right, no amino acids, smaller peptides. But carboxypeptidase A and B comes from the pancreas, and it's an exopeptidase. The pancreas does not contain, well, you can cut from either end, right? Carboxypeptidase, you're cutting from the carboxyl end. Aminopeptidase, you're cutting from the other end, okay? Well, the pancreatic enzyme, we don't have an aminopeptidase, okay? We do have an aminopeptidase in the brush border, which is not. At any rate, you're going to have, what you're going to have is you're going to have amino acids being generated, okay? It makes sense so far?
Yes.
Yeah. Now, the end result of all this, just review again, is you're going to have some ditripeptide, larger peptide, amino acid. When someone brings up ditripeptide, that means it might be important, right? Why am I focusing on ditripeptide and then lump everything else into larger peptides, right? Well, I'll tell you in a minute. Amino acids, well, we know this. All right? In the brush border, we have peptidases, right? They can take whatever peptides you have left, no matter how long they are or whatever. Again, you're going to make ditripeptides. And you're going to make amino acids. Now you get in the picture? We have transport for amino acids and for ditripeptides, but nothing larger than tripeptides. But we can transport ditripeptides. Remember, carbohydrates, only monosaccharides, right? Single unit. Here, you can a single amino acid and ditripeptides, okay? And if the ditripeptides get get transported in, inside, remember in the first slide I showed you, inside the cytoplasm we have a variety of peptidases that convert everything to amino acids. That result in, you take protein, what comes out in the portal vein is amino acids, right? This is a transport mechanism. Remember SGLT1, sodium glucose cotransport? Not a problem, we know that one. SGLT1, but we don't have a name for this. There must be 7, 9, 10, I think the number is growing, of these amino acid transporters. So they don't know who to blame it on. They used to say, this is a basic transporter, then they have a third one down here, neutral, acidic. But it turns out they're all promiscuous. What that word means is they will transport whatever's there. They may have a preference. So some textbooks still use basic neutral acidic transporters. Remember your amino acids? You had side chains based on the side chains. They were basic, neutral. Anyway, the point being is that we don't have a name for these transporters. They're just sodium amino acid transporters. There's no name for them. And because of that, one reason is that we don't have that many nutritional deficiencies. We knock out one of these 10 transporters, the other transporters pick up the slack, if you will. Okay, can be transported another way. Let's say you knock out a real important one for, let's say, glycine or something like that. We still don't develop a nutritional deficiency. Now you will in the kidney. I think it's called cystinuria. They have a transport problem in the kidney, but that's a kidney issue, that's renal. But the other thing that helps is peptide transport. Now here you have to do a little memorizing. This is the transporter, peptide. Don't tell me it's going to be hard. Peptide transport, peptide. You'll remember that, right? What's going to be tricky is to remember that it's proton driven, not sodium, hydrogen driven. Okay? So, again, you can start out sodium ATPase, you're creating a gradient. You need the proton to move di or tripeptide. You need that proton. So you generate that proton using an exchanger. It's called a sodium hydrogen exchanger. Okay? It's all written up here. So, you got the sodium gradient, hydrogen, it's recycled. Okay? Just keep going. As long as you have peptides, all right? Is that okay? Now remember when I said about exchanges, you have to take a guess at someone. Someone says you're moving hydrogen one way, you're going to be moving chloride the other way?
No.
No, you have the same decision. That's right. So take a guess. If they give you choices, you already know predominantly on an exchanger, you're going to try to maintain electrical neutrality and everything. So if you're moving chloride one way, it's got to be a negative one, probably bicarb, right? You're moving hydrogen one way, what's the common one? Well, it could be potassium, right? Remember the hydrogen potassium ATPase in the stomach? Okay. So, is this okay? We've learned quite a bit, right? Is this okay? This is a summary slide, which you will love, I'm sure, right? You want to memorize it? You already know it. You've already memorized it. You can use this to study from, or study from all the other slides that we created. Let's take a look at it. Gastric, pancreatic peptidases, they lump them. Yep, don't forget you have a carboxypeptidase in the pancreas, it generates some amino acids, right? And I mean, this is AA, amino acid, and means make up a chain length, right? N number, how many you got to hook together, right? And in this case, let's make it 5, okay? Okay. All right. Okay, so you have some amino acid and oligopeptide. Amino acid, we know this, right? Amino acid. Hey, there's a transporter for amino. One of many. That's good, right? One of many. And it uses sodium, right? Gets into the cell. No problem. What happens to the oligopeptide? Oh, we know we have peptidases in the brush border. A lot of them. And what does it say? One of many. Okay, so now you got this, what's a 4-peptide? Quadripeptide? I don't even know what the name for it is. Let's say 4 amino acids, right? You run into a peptidase. Okay, now you got 3 amino acids. Now you got a tripeptide. Well, guess what? You have a transporter for that, right? Oh, good. Except it's using hydrogen. You transport it to the tripeptide. Let's say you didn't transport the tripeptide. You missed the transporter. Whoops, motility or something. We got another peptidase here, right? It's going to take it down to 2. Now you go, oh, we still have a ditripeptide transporter. It's going to bring it in. It's going to use hydrogen. Let's say somehow you missed this one. Well, you have another peptidase that takes the dipeptide and turns it into amino acid. Bring it in. We already know this, right? Now inside the cell we have peptidases. And here all they did was say you got 3 amino acids here, tripeptide. Well, we'll use a tripeptidase, amino acid. You got 2 amino acids, dipeptidase, 2 amino acids. Unlabeled. They don't know what the names of these things are. Facilitated diffusion out. Was that a review?
Yeah.
Is it as hard as it looked in the beginning? No. But if you don't have to know this for the exam. It's your choice, because the same material was presented earlier, right?
Sorry, you mentioned the gas.
Huh? The what?
You mentioned the gas. Review.
The what?
The gas.
Gas. This is protein we're talking about,
Yeah.
not carbohydrates. You don't know that much about what bacteria do with protein.
So
Carbohydrates is the main issue, cellulose, fiber.
So it's still on the research. That's what you're saying.
Maybe if someone's looking at it, I don't know. I don't follow that. You can look it up. I'll tell you what, next class, we'll report about it.
Yeah, if I can find anything.
No, if you're finding anything, I wouldn't mind hearing about it, but so does that. Let's look at lipids, and here we sound simple, but it's not. Here we're only going to be dealing with the pancreatic and attendant. Well, bile salts is a big one. I think you covered that when you talked about bile salts and how they help with lipids. All right, let's go into that. Lipid digestion hydrolysis. Okay, here I have in red, what? This is triglyceride, right? Is that what you're used to hearing?
Yeah.
Yeah. Have you ever heard of triacylglycerol? In biochem, your lipid class, when you get to that, it's going to be triacylglycerol. It's changed. This time I don't know the reason, but it's changing. But again, if we put this somewhere in MCQ, or if you're taking biochem, if you're doing biochem, don't say triacylglycerol. Okay. If you're doing some, still some textbooks will say triacylglyceride. Okay. But it's changing. Just like I brought up with enterokinase, entropeptidase, be aware, it's changing. But mostly, all the recent biochemistry books deal with it as triacylglyceride. but some of the physiology and anatomy book stuff haven't caught up yet, okay? But that's what you're gonna get. We'll just call it triglycerides for now. All right, and, okay, long chain versus short chain. Okay, I made this number up, but not really. You have to have a cutoff somewhere. Oh, you haven't had lipids. Have you had, you know that the longer the carbon chain, the less water soluble it is?
Yes.
Well, where'd you get that? Isn't that lipids? Well, as long as you got it somewhere. The shorter the carbon chain, the more water soluble, right? Okay, that's all you have to know. And somewhere you gotta decide what's long and short. So you take a pick. And most of the time it's between 12 and 14. I even have a dash here. I didn't pick a number. 12, 14. Some people say the cutoff is 16 carbons, okay? Then you're dealing with a long chain. But all they're trying to say is long chain is not going to be soluble in water very well. Short chain will be, medium, so-so, right? That's all they're trying to say. So your salivary and gastric lipases, they deal mostly with medium and short chain. That's the preferred substrate. It doesn't mean they won't hydrolyze a long chain, but they will, but they prefer a short and medium chain. And they generate diglycerides, which is, you know what triglyceride is? 3 fatty acids and a glycerol. Diglyceride, 2 fatty acids. Monoglyceride, 1 fatty acid and a glycerol. Is that okay? I hope we got that. All right, they'll generate diglycerides, okay? They're acidic, but they only are responsible for about 10%, 15%, some people have 20, of total lipid digestion. Most of the time, everything is going to happen in a small intestine, and your pancreatic enzymes are key to that. I have a little asterisk there. Pancreatic insufficiency. Now, you may not know what that word is, but you know what sufficient means?
Yes.
If it's not sufficient, it's insufficiency. And usually it can destroy your pancreas. I hate that word, destroy. Well, it is. It's destruction, like pancreatitis, be it from cystic fibrosis, or they have a lot of alcoholic pancreatitis in the West. But you destroy the pancreatic tissue. You have to destroy 80% or more of your pancreas before you affect digestion in the small intestine. That's how much of a reserve you have of these enzymes. Pretty good, huh? We're ready for that food. But anyway, it's a problem. If you have pancreatitis, then these gastric lipases become very important. Because you don't have the pancreatic lipases anymore, right? And on top of that, the preference for short chain and medium chain, you can change the diet for that person. Keep them away from long chain, right? Newborn, most of your milk fats are going to be medium short chain milk fat, okay, and that's what newborns are drinking. On top of that, their pancreatic enzymes, they're still developing. They're not up to full speed. It takes them a while for all the pancreatic enzymes, the whole gamut to be matured and developed, etc. Then they take over. But they're more dependent. So a neonate, young, just born, will be more dependent on gastric lipase than they will on pancreatic. Makes sense. Anyway, the pancreatic lipase, most 89%, we know about their pH optimum, more neutral, and we're thankful that the pancreas, and even the liver, remember the liver, hepatic gut cells will secrete bicarb as well, right? Is that mentioned? Yeah. All right, so you're making sure that the pH is at the level. They even go down the same duct, usually, right? Common duct into the intervoting pancreatic enzymes and the body, right? They even enter through the same duct, which is nice. So they're trying to keep the pH at the pH optimum. Long chain triglyceride is going to be that's what bile salts do, emulsification and solubility. We'll cover that. Emulsification. You know what emulsification is? Okay. This is from Guyton, very good. Fat, emulsified fat. How you do that? Bile agitation. All right, I'll give an example real quick. Have you ever done dishes? But if you have a good meal, nice, good, tasty, most of your taste is in the fat component, lipid. So if you put your dishes in the sink and you go, I'd rather watch TV, I'm full. You come back an hour later, there's going to be an oil droplet on top of that water. You ever seen that?
Yes.
Or, okay,
Yeah.
you go, oh, I don't feel like doing dishes yet. You'll mix up that top, and all that oil will break up into little pieces, right? You walk away again. Come back another hour, it's back again as a big oil droplet. I mean, that's called, you emulsified it by shaking it up. In a GI tract, we emulsify by motility. When you have contraction, what do you think you're doing? You're mixing that stuff. In a stomach, you're really smashing it together, right? What do bile salts do? They're amphipathic, right? So they're hydrophobic. This is a cartoon. I'll show you a structure, but this is not biochem. Hydrophobic and hydrophilic. You know what that means? Amphipathic. They can interact with both. So this part will usually interact with the lipid part, this with the aqueous medium. So you got a really nice thing. What bile salts do is the same thing as a detergent. When you're washing dishes, you'll notice most people use a detergent. You don't need a detergent to wash off carbohydrates or protein. So you need a detergent to help you keep the lipid and small particles emulsify to keep them washing away. Bile salts are endogenous detergents, if you will. They're amphipathic. And what they do is, as soon as you make those little droplets by shaking or motility, the bile salts surround them, right? They're interacting with the lipid portion, big, small, smaller, and this hydrophilic side, they're interacting with the aqueous. So they're preventing these from coalescing, coming back together into one big lipid droplet. They bump into each other, right? No problem. So what they do is they prevent, right? Does that make sense? Coalescence of the lipid. The motility is going to break up. That's what emulsifies. The bile salts prevent it from coming back. So by emulsification, you have created from one huge fat droplet, let's say it's 1,000 micron, you generate a whole bunch of 1 micron small fat things. They're surrounded by bile salts, right? Call them triglycerides for now. And here you got motility, bile salts, and lecithin. That's still used in textbooks. I think biochem will switch you to phosphatidylcholine. Lecithin is a phospholipid. Egg lecithin.
Do bile acids repel each other?
I'm not sure if you want to use the word repel, but they prevent, because they're surrounding the lipid doesn't merge together. Either way you want to think about it is okay with me. Because you've got the right concept, you've got the right idea. What the physical chemical interactions actually are, I don't know, and I don't want to mislead you. I don't know what the result is. But they keep them emulsified, is what it is. Or prevent, do I have it? Yeah, coalescence. Okay, so what have you done by all this? I don't know if you know this, you take something large and make the same volume, but make them into smaller, huge surface area increase, which is nice, because lipase is what's going to digest the lipids, right? We have phospholipase as well, and they have another one I'll show you. But these are proteins. These enzymes are proteins, right? They're water soluble. They can only get to what we call the lipid aqueous interface. They don't go inside. The lipase can't get in here and start cutting because it's not lipid soluble, it's water soluble. So it has to position itself on the surface of these little fat droplets to be able to digest the lipid. Does this make sense? Because it's water soluble, right? All right. So that's why increasing the surface area increases the area over which this lipase can act. Otherwise, we always have steatorrhea. Right? You're going to remember that word, steatorrhea. Huh?
Yeah.
Oh, yeah. All right. Here I said triglyceride, but you know it might change to triacylglyceride, but that's okay. Now you have bile salts surrounding this, right? Aqueous. The lipase has to get in here. How does lipase get in here? It helps. It gets help from coli. We'll get to that in a minute. What does lipase do? It converts triglyceride to a monoglyceride at the 2 position. The way it's written here, I think 2 monoglyceride sounds like it's generating 2 monoglyceride molecules. It's not. But when you get to biochem or see, position 1, I have it on the slide, how about that? Position 1, 2, and 3. So position 2, they don't attack that particular ester bond. pancreatic lipase. They'll attack at the 1 and 3 positions, right? So you're going to get 2 fatty acids and 1 monoglyceride that has a fatty acid attached to the position 2. That's all they're saying. Is that okay? It's hydrolysis, right? You cut off fatty acids. All right, a little bit about colipase. It's also secreted by the pancreas, and it's activated by trypsin, as you can imagine. Gastric lipases don't need this particular colipase, so don't worry about that. It's only important in the intestine. Why is it important in the intestine? Because you have bile salts. In the stomach, you're not going to have bile salts. This is an experiment. You guys, some of you guys might be scientists. I doubt it. You want to be clinicians, right? But anyway, let's say you're a scientist, and you put a fat droplet in a test tube, and you're going to add some lipase, L, to this fat droplet, and you go, is it working? Well, one way to do that is measure fatty acids showing up, right? If you're getting fatty acids, it's working. It's cutting, right? Makes sense. So, you put the lipase in, it attaches to the aqueous lipid interface and generates fatty acids. You add bile salts, you emulsify it, all of a sudden you're not generating fatty acids anymore. Even though you emulsify it, okay? You're not generating anymore. Because the lipase is pushed off and it can't get to it because of the bile salts. It can't get to that aqueous lipid interface because the bile salts are surrounding it all. And bile salts are amphithetic. They can surround the whole thing. Lipase, unfortunately, is just water soluble, right? So now you add colipase. What colipase does is it binds to the lipase, 1-to-1, and anchors it to the bile salt lipid interface. So you got the best of both worlds, okay? You've got it emulsified. You're keeping it emulsified with the bile salt, and colipase is allowing lipase to keep eating, and that's why you get fatty acids going up again. Anyway, just what colipase does. At any rate, what you're going to get is this is now we're talking about micelles. Now, first thing, I think Mohammed covered all this, emulsification, right? We covered it. The other important thing is formation of micelles, okay? Now, micelles, bile acids, I think you covered that. Bile acids form micelles all by themselves. In the gallbladder, everything is micellar. That's how you get stones only if you have less bile salts or more cholesterol, more lipid-soluble material. You come out of solution, and you get stones. But in the gallbladder or in bile, they're going to be micelles. So they're usually at what they call critical micellar concentration. You put 2 bile salts together, they may not form a micelle. But if you have 4 or 5, all of a sudden, you know what a micelle is, or a liposome? No? In other words, this is a micelle. But in the gallbladder, they may not have fat or monoglycerides in here. They may be holding cholesterol or some other lipid in the middle. Okay? But they don't just float around. They form micelles. Micelle, huh?
Even in the gallbladder?
Yeah. Otherwise, you'd have lipid on the surface. Cholesterol is insoluble. Doesn't cholesterol end up in the gallbladder?
Yeah.
Yeah, it's insoluble. It's fat soluble. It's a steroid. Anything with a steroid nucleus, it's not very soluble. Anyway, don't worry about that. But when people use liposomes, we've created them in the lab. All we do is we just use phospholipids and stuff like that, and you just put it into a homogenizer and you got liposomes. Put whatever drug you want in there and deliver it to the intestine for absorption. Drug companies do that. You'll hear about that. They deliver some drugs with liposomes. Lipid-soluble drugs they deliver with liposomes. You've got triglycerides. You're forming fatty acid and monoglyceride. And now you've got micelles. These are small. You've got plenty of bile salts. They create micelles now. As you're coming off, bile salts interact with these, creating micelles. These are very small. Remember, this last one was 1 micron or something. Now you're approaching 1 ton. These are so small that they're in solution. You actually put the lipid material that you've generated into solution. Okay? You can. The emulsified solutions will look cloudy. Have you ever seen emulsions or something in test tubes? They look cloudy. But if you have a micellar solution, it's clear. You can see right through it. That material, you've just now took this big fat dropule, using lipase and bile salts, and made it into a clear solution. Okay, soluble. Which means it can get to the enterocyte now. All right, can get across that aqueous medium. And that's what they think that my cells are for shuttling fatty acids to the enterocyte. This is a cute cartoon from a textbook. The GI, physiology, doesn't matter. But this one shows fatty acid absorption in the presence of bile salts. You see these micelles? Okay? And fatty acid in the absence of bile salts. She's telling you a true story. It's not all or none in biochem or physiology. And okay, you can have a long chain fatty acid, which we do here, okay? And you can put 1,000 of them into a beaker. One out of that 1,000 may go into solution. You have a shorter chain fatty acid, put into solution. More of them will go in solution, but some of them won't. It's all relative, okay? Now, you can get some lipids, or in this case, we're talking about fatty acids, that's the symbol for fatty acids. Some of them will actually diffuse and make it to the enterocyte. But this is what I call the effective concentration. One, if you have bile salts, 1,000,000 times more will be moved to the enterocyte. And this is what you see, what basically everybody, the way you have it as a cartoon is my cell comes up here and puts fatty acids into the enterocyte, right? But here they show you it's disintegrating, you know, as it's coming up. And the bile salts, they're not going to be absorbed here, right? We know that. But the effective concentration, what you've done is delivered these fatty acids to the epithelium, to the plasma membrane, at a huge concentration. That's why they call it a shuttling system, right? And if it helps you to have a cartoon of a little ball carrying everything and opening up and dumping, that's fine. It's the same concept, right? You're delivering more of something insoluble because you solubilize them in micelles. Drug companies do it with liposomes. Your body does it with micelles, right? All right, is that good? And that's why I have effective concentration, 1,000,000 here to 1. You're going to get 1,000,000 fatty acid molecules being absorbed as a plasma membrane with micelles. You may get 1 or 2 being absorbed without. The bottom line is most of these lipids, if you didn't have micelles, are going to end up where? If you don't absorb it, you end up in the colon, and then from there, if you don't have bacteria to ferment it, you end up in your stool. And it's used clinically as diagnostic, called steatorrhea. And one of the reasons it's used is patients will smell it. It's very malodorous is the word. I think I mentioned this before, right? Yeah. In other words, you'll be going to a physician, don't worry about it. Bile salts, they're not absorbed, recycled. It's called the enterohepatic circulation. You have that, right? Short chain fatty acids. They don't need micelles. They can go into solution. So you can put them over here. In the absence of bile salts, a lot more short and medium chain fatty acids will get in. They don't need the micelles. You usually don't have a problem with short and medium chain fatty acids or triglycerides. Is that okay? Yeah. Oh, 10. We're almost done. It's reminded me, this is not step 1, step 2, step 3. This is all going on at the same time, okay? The bile salts are always there. Let me put it, let me give you a, just work. Oh, that's how it turns out. Okay, let's pretend you don't have monoglycerol or fatty acids. These bile salts on their own, the hydrophobic parts will interact with each other, hydrophilic parts, they're going to form micelles all by themselves. That's the way they are in the ball. They're going to form. So you have micelles already there. They're called bile salt micelles. When you start putting in fatty acids, monoglyceride, and we'll see later anything lipid, they're going to be called mixed micelles. Okay, because they have a mixture of everything lipid soluble. So you're going to have bile salt micelles at the same time you're having hydrolysis. Okay, you've got these bile salts around the fat, you're going to have micelles they're sitting there. That's the way they are in aqueous solution. So as soon as the fatty acids come off, you have a bile salt micelle sitting right here. It's not like some new bile salts came along and made it micelle. It's all there. And the way they just get incorporated into this structure. Does this make any sense? No? I guess, did I make it worse by explaining that?
No.
Sometimes when I take a look, I go, oh, did I say, maybe I shouldn't have said anything. But they're there all the time, okay, and they're just using, and it's a shuttling thing. It helps them get to the aqueous end. That's why I like this one. It doesn't show my cell going up here and just dumping everything. It's a process. It just delivers. It's a way of shuttling fatty acids to the enterocyte. Do we cover this okay? All right, there's other things that we can attack. Get are going to destroy mineral salts as well. Anything lipid soluble, right? Let's talk about that. We talked about this, triglyceride. You have cholesterol ester. You can ingest cholesterol as a cholesterol, but usually you inject it as an ester. That's what that is. That's a fatty acid attached to a cholesterol. Okay. You have lecithin, or if you want to, phospholipid. Same thing, depending on what book you're reading. Phospholipid is probably more appropriate. And the major one in your body is phosphatidylcholine. Have you had any of this about lipids and stuff? You'll get that. You'll get it. Phospholipid. Okay. At any rate, you have phospholipid here, or lecithin. And you'll watch these enzymes. Remember pancreatic lipase? It takes off 2 fatty acids, and you have a monoglyceride, right? Cholesterol ester takes off a fatty acid. Then you have cholesterol. Phospholipase A2 takes off a fatty acid. And you got lysophospholipid, lysophosphatidylcholine, lysolecithin, whatever you want to use. But you notice every one of them is taking off a fatty acid. The triglyceride, you took off 2 fatty acids, okay? All right.
No, I just.
No. All right, all these substances are going to end up in your micelles, right? All of them are. They're going to end up in some place so they can be protected from the aqueous environment. And now we're going to be starting to call them mixed micelles, not biosalt micelles. The biosalts can make micelles on their own. They don't need anything else. But mixed micelles, that's what they're referred to. The other thing that's going to go into a micelle is, guess what? Anything lipid soluble. Yeah, I put vitamins here because they're pretty important. But anything that's lipid soluble. If it's not soluble in water, it's going to find those micelles. Yeah.
What's exactly the role of cholesterol ester?
What exactly do
The role
they?
of cholesterol ester. The function. What's the difference between the cholesterol ester and
These are more hydrophobic. These are more empathetic. They'll be able to get across membranes and stuff and deal with the aqueous interface. Technically speaking, though, even with cholesterol, they now have found a protein. As with all these lipids, they get into the cell, and guess what? The cytoplasm is aqueous, right? They have proteins that shuttle them. But that's getting beyond this stage, okay? But they'll have proteins that shuttle them to the smooth endoplasmic reticulum for resynthesis. But yeah, some people even think there's a transporter for cholesterol in the membrane. So you go, okay. But do you need to know that? If you want to, we can talk about it after class. Or email me, I'll send you a reference or something. But for now, I mean, we're getting way beyond what we have to do. And for medical students, if you're a graduate student and you're going to get involved in this and research, then you'll be reading papers and stuff on the more recent trends. But in this case, I brought up vitamin. You have to memorize this. Do it any way you want. You know, use an acronym. DAKE, DEEK, whatever you want to use. These are lipid soluble. If you buy them, don't even tell you to take it with a meal. You know. But if you ever seen vitamin E, they're coming in capsules. It's a gel, gelatin, oil. Like with any oil, you got to solubilize. You solubilize myself. So if you don't have bile salt, you're going to get diarrhea. That's true. You're not going to absorb triglycerides or anything else end up in your stool. So you're also going to have a problem with lipid-soluble vitamins. They're going to pass right through. They need to go through the micelles to get absorbed. Okay? They're not digested or hydrolyzed or anything like that, but they need to be solubilized and be taken in. All right, so what do we do? You took out all the fatty acids off these things. They're going to absorb it, and guess what? You're going to put the fatty acids back on. Okay. That's all you have to remember, all right? And this slide is showing that. These are using different symbols, but don't worry about it. This is your micelle, right? Cholesterol, monoglyceride, lysophospholipid, fatty acid, right? We had that from the previous slide, right? They're going to get into the cell by diffusion. Cholesterol, and as I mentioned, it may be transported, but let's not worry about it. Cholesterol, monoglyceride. And look what happens to the fatty acids. You're just going to add the fatty acids right back on. You're going to end up with what? Cholesterol ester, triglyceride, phospholipids. So basically, the pancreatic enzymes took them apart. The enzymes dealing with resynthesis and which endoplasmic reticulum, right? Which one? Smooth. Remember? You guys have this? Smooth and rough? Yeah. Smooth endoplasmic reticulum. You're going to resynthesize. You're going to put them right back. Okay. It looks complicated, doesn't it? But once you understand, you took it up, you're going to put it back. Okay. So it's called reesterification, if you want the name for it. And then, now you've got this lipid droplet inside of the cell. So basically, you took a lipid droplet from outside the cell, put it into your smooth endoplasmic reticulum. That's the end result, right? But you don't want a lipid droplet inside your cell, which is growing. You got to get rid of it, right? So what you do is you have to help it be transported in aqueous media, your blood. Now, you haven't had lipoprotein yet, like I got it from the other class, lipoprotein. And chylomicrons are probably the biggest lipoprotein. They're called VLDL, high density lipoprotein. Have you heard of these things?
Yes.
Yeah. And chylomicrons are going to be the biggest one and the least dense, because they're always going to have triglycerides. The other ones become more dense because they'll have more proteins around it and more cholesterol in the middle. That's the way you transport fat in your body, in your blood. You've got to get this lipid to your cells, right? Otherwise, why are you absorbing it? Just for kicks? No, you're absorbing it because you need that energy. You haven't had beta oxidation yet, right? No. You will. That happens in the mitochondria, and it generates a lot more NADH. Do you have electron transport?
Yes.
NADH goes up in the complex 1, you generate ATP, right? Well, you're going to get a lot more NADH from beta oxidation than you will from carbohydrates. That's why they call it a high energy lipid, yeah, high energy. Twice as much energy per weight than a carbohydrate. You could recover by beta oxidation. That's happening. Never mind. I'm getting away from this. But anyway, you've got cholesterol, triglyceride, phospholipid. Now you've got a lipid droplet again. And on rough endoplasmic, you got to generate a protein. Proteins is going to help surround it and help it interact with an aqueous medium. So the protein, they have hydrophilic side chains, right? And these are called apoprotein, and the one for chylomicrons is ApoB48. You'll know what it is, right? I'm not going to give you a trick quiz like it's an ApoB40, ApoB30, I don't do that stuff. But I might say apolipoprotein or apoprotein, something like that. But anyway, it's on the outside. And phospholipids are also very amphipathic. They're going to help solubilize it. So now you got this big fat droplet, and you go, oh, okay. You have to get it out of the cell. They usually do it by exocytosis. Did you ever hear of that? Yeah. Exocytosis. And because they're big, we'll cover that in cardiovascular, the intraendothelial junction won't let it get through the endothelium. So here they are stuck in the interstitium forever and ever. No, they go into the lymphatics. Lymphatics have wide open spaces between their endothelium. Everything that gets into your interstitium is going to get into your lymphatic, including bacteria. That's why you have lymph nodes everywhere. They crap that bacteria that got in, let's say by accident. Because bacteria can't get into your bloodstream. Okay? But you need it delivered to your other organ systems. Where do the lymphatics go? You guys cover that?
Yes. Inferior vena
They go into the thoracic stuff, the subclavian. Yeah, right. So they end up in your blood. Yeah. And as a matter of fact, if you take if you're taking blood samples for whatever reason, sometimes you tell the person to fast, if you're eating a lipid meal, and you spin it down for you haven't had hematocrit yet. You know what hematocrit is? Huh? Doesn't matter. You'll have a huge what they call Buffy Coke, which is your lipid when you spin it down. It spins out. Don't worry about it. Anyway. All right, is this okay? Goes into and back. Somewhat, right? All right, just like before, just from boron and bullpen. Remember this? You hated the amino acid one, right? Or the protein. This is the same thing we've just been talking about. Let's talk about it again. Let's make it bigger. And you notice that it's another summary slide, so don't worry about it. If you don't like it, use the slides before, okay? Now, well, you know this is an absorptive cell, right? Epithelium. Okay, we'll put it here. We've had the mixed micelle. It's got everything in it: cholesterol, lysophospholipid, long-chain fatty acids, monoglyceride, right? This unstirred water layer, think of it as your mucus layer. It's not stirred very well. You know, and you're trying to get this material to the enterocyte, so you need micelles. They bring it here. Where is one? Okay, now they diffuse in. And see the arrow? Smooth endoplasmic, right? SCR, smooth endoplasmic reticulum, long chain fatty acids, another. All right. Number 2, you're resynthesizing them, right? Reesterification, putting on the fatty acids. Or today they're using the word converted back. All right. Fat droplets up here. Then where do they go from the endoplasmic to the Golgi, right? They're packaged in the Golgi. All right. Apoproteins are synthesized in any protein. I mean, a rough endoplasmic reticulum. All you have to do is read this stuff. Apoproteins is, okay, that's 3. Where's 4? 4. Chylomicrons arrive at the Golgi. And what I've crossed out is VLDLs here and glycosylation process, because you don't have to know that. You don't know it yet. The chylomicrons is fine. And now you take the you know what happens at the Golgi? They butt off, form a vesicle. Is that right?
Yeah.
Remember that? And there you go. There's your chylomicron, exocytosis. And every step is written out here. Okay? So if you don't like this, don't use it. But from past years, once they understand the lecture, the students, when they were doing, they just grab that slide, they go, oh, I'm good. But you may want to. Is this okay? Yes. You want me to go over it again? We don't. No, I'd say no too. I agree with you. One last thing, I have to cover it. Vitamins, we already covered this. Where the vitamins going to end up? They're going to end up in your chylomicron. That's the way you're going to transport lipids out of the cell. And it's going to go into the lymphatics, not into the bloodstream, because they're just so huge. They're big. Vitamin C, water-soluble vitamin. And a lot of the B vitamins that are water-soluble, except for B12, which we'll cover at the bottom. They're usually using, again, sodium co-transport mechanism to get across, because they're water-soluble. If it's water-soluble, it's going to have to use some kind of mechanism to get across that membrane. All right, and with vitamin B12, since we brought it up so many times, intrinsic factors, things like that, this is the way it works. At the ileal enterocyte. So when you get down to the ileum, what else happens to the ileum? Bile salt, active transport. You covered that, right? That's part of the enterohepatic circulation. And you'll also have this transport. And this is endocytosis. But they take up the complex, the vitamin B12 intrinsic. They take it up. And they protect it. When you have transcobalamin, by the way, cobalamin is just another name for vitamin B12. I don't know if you knew that or not, but some textbooks will just say cobalamin. What they're talking about is vitamin B12. Transcobalamin is going to transport that. Okay? Any questions? Are we good? We have an early run. Oh, wait a minute. What about attendance?